I'm Allison Watson. I'm co-founder of Ring20 Research and Support UK. And I'm here today to talk to you a little bit about the rare disease I represent, the patient organisation, who we are and what we do, a little bit about our current research strategy and how genomics might actually fit into that. So I'll kick off by explaining a little bit about what ring chromosome 20 syndrome is, because this may paint a picture for some of the challenges that we have. So Ring20, for short, as we call it, is an ultra-rare disease. The main symptom is epilepsy that's refractory, doesn't respond to any treatments. It's associated with behaviour difficulties and cognitive regression after the seizure onset, because interestingly this disease does not present at birth. The seizures typically start somewhere between the ages of 4 to 10 years of age. Interestingly, 1 in 50,000 live births are reported to have a ring chromosome, and yet there are only 150 cases of Ring20 cited in medical literature worldwide. So we're that rare. So for that reason, it's perceived to be underdiagnosed and under-recognised. We actually believe there are a lot more patients out there, just haven't been detected just yet. And it's likely that they're either going undiagnosed for some of the reasons I've kind of alluded to, or being misdiagnosed, particularly either in the epilepsy field, being given under an umbrella epilepsy diagnosis incorrectly, or in some cases psychiatric and not recognised as having seizures. Challenges that we have, I think I kind of mentioned this, is diagnosis is difficult. The standard testing doesn't pick up the ring chromosomes, particularly in the mosaic form, where there isn't any change in the actual genes to pick up, which is what those techniques are looking for, in my layman's terms. Ring20 can present in the mosaic or non-mosaic form, so non-mosaic being in every chromosome 20 in the body, mosaic being in only a few of the chromosome 20s in the body. And in the mosaic form, it can present in as little as 5% of the cells. So when they're running a karyotype, which is the only way to see the ring through the microscope, you need to look for 50 to 100 cells. Now that's important because typically the neurologists or the paediatricians submitting the tests don't usually send that many cells. So again, difficulty in not getting the diagnosis. We're so rare, so little is known that there's no recommended treatments for the disease. There are no clinical practice guidelines. The doctors are working blind, treating on a trial and error basis. There's no patient registry just yet, but we're working to change that. There's limited research, both clinical and genetic, and there's no natural history studies. We don't know what the future holds for our patients. We've got no biomarkers. But I'll turn that on its head and say, okay, with great potential for research, come on, guys. And we're really interesting because, as we said, Ring20 doesn't typically present with anything different at birth. So you don't see any traits, no medical learning behavioural problems until the seizures start at 6, 7, 8 years of age. So you don't think that there's anything wrong. A little bit about us as an organisation. We were formed just about 5 years ago. We're very much a very small voluntary organisation, run by myself and our chair, Don Gordon. We are actually the only patient support group in the world for patients with Ring20, so any patient that can speak English or can communicate with us through Google Translate will connect with us. We've recently established a medical advisory board, which is helping us with our research strategy. And we're supporting just over 100 families worldwide currently today, which is not bad given the number in medical literature. And what we do, in a nutshell, primary aim is vital support network for those patients and families, giving them some kind of mutual support between each other. We raise awareness of the condition because talking about it might lead us to finding new strategies. We provide support for the healthcare professionals they're running blind, so we try and signpost through our website to latest information, trusted information sources, and we're now trying to raise funds for vital research and support any research that might be happening out there or that we can kick off. So our research strategy, so we've been raising funds for the last couple of years, and we hope sometime later this year, if we can get our proposal off the ground, we will actually get on board a natural history and biomarker study, which will actually give us some traction. And as part of that, we want to start a patient registry, which is fundamental to getting involved in future research. Alongside all of that, we've been talking to the NIHR Bioresource Unit and talking to them about how can we and other rare disease patient groups create a patient-led pathway to suggest projects to engage with them. So we're trying to reach out beyond our small horizons. And we've been working this year with Patient-Led Research Hub from Cambridge to drive, or we've even initiated patient-led research, conducting a literature review and a survey on the use of ketogenic dietary treatment, which is potential treatment for rare epilepsy that might be more beneficial to our patients. And we've taken that one stage further, because what I haven't mentioned is I'm also a patient advocacy representative. I'm the co-lead for Epicare, the European reference network for rare and complex epilepsies. So I have in my small role as Ring 20, I have a much bigger role in the European space, representing all families and patients in that space. And within that role, I have the opportunity to work with the healthcare professionals within Epicare, and we're looking at taking that patient-led research to see whether we can move that towards an observational study into use of KDT, either within a Ring 20 cohort, if it's large enough, or a wider rare epilepsy cohort. So we're very open to collaboration. And again, working with Epicare as an umbrella to try and reach out to a wider audience to raise the awareness of the diagnostic challenges and how we can overcome those. So, highlighting there that we're looking to introduce a patient registry. So if we're going to be doing that, we need to be thinking about what kind of level of genetic or genomic data we need to incorporate within that, and that's part of my learning here today. So I had to go and do my homework, because I'm a layperson, and you have to forgive me. So I had to Google the difference between genomics and genetics and get it right in my headspace. And the World Health Organization popped up, and I saw that genetics looks at the single genes, whereas genomics looks at the wider interrelationship of the genes and how that influences the growth and development of the organism. And I thought, interesting. Genomics, Ring 20 is a chromosome mutation. Currently, there's no single gene, or we don't know which genes are implicated. It's hypothesized that the ring formation is causing a difference in the gene expression, and that's what's causing the seizures. So perhaps is genomics more the pathway to finding the answers to, why the seizures are caused, rather than looking at the specific genetics. There is some genetic research happening in the US over at CHOP. They've been looking for a number of years at ring chromosomes and gene expression, but that's not something that we're driving or that we're funding. So we're recipients of their data and the outcomes of their research, rather than participants directly. So in terms of incorporating genomic research into our research strategy, I looked up again, I did some homework, looked at, what opportunities could this bring to us? Well, integration could bring earlier detection of a disease. Well, that would be fantastic, because if we can detect the disease earlier, and if we can get on top of the treatment earlier, we could potentially improve the outcomes for patients. So that's really important. We could increase diagnostic rates. I think that kind of speaks for itself. But actually, that would turn us perhaps into a larger cohort of maybe a few thousand rather than a few hundred, and that would perhaps make us more attractive to researchers and for future clinical trials, because there's never been a clinical trial in Ring 20 to date. And if we could find some answers to improving treatments, then ultimately we improve quality of life. But the challenges that we face, the big challenges that I think we face, are around this ability to detect the rings. If we can't diagnose the disorder, then we're almost stopping at point one. So we need to overcome that in some way and try and find out what we can do to get that diagnostic rate improved. We need to do more to understand what's going on with the gene expression. And ultimately, we can't do this alone. We're a tiny patient organisation of a few volunteers with very limited capacity and limited funding. You know, we need to be clever. We need to know who we can partner with to take things forward. And how we aim to do this? Well, constantly raising awareness, talking to people, finding out who we can work with, coming to events like this today, learning and understanding and seeing what we can achieve, and ultimately collaborating. Because I think if we look at individual rare diseases on their own, 6,000-7,000 diseases, and we're all trying to do things individually, it's going to be a long time to get anywhere for the really ultra rare. So working together, perhaps we'll make a ways forwards. And that's it from me.